Hey team, Dr. Jack Gordy here, and in this series of videos I'm covering the problems with science. And so in this video, I'm going to cover something called translational failure. So let's jump into it. In the previous video, I covered some of these scientific 101s, these fundamental principles of science that I think is really critical that everyone understands. First of all, the placebo effect, which is kind of merely thinking you're getting an efficacious treatment will change your perception of the disease and might actually make you feel better, feel like your disease is subsiding. And so there, when measures are subjective, there are very large things that can be influenced by the placebo effect, your expectation of treatment. There's observer bias, which is that subjective measures measured by scientists and doctors can be influenced by the expectations of the results. So if I'm a doctor and I think your blood pressure should go down, and then I measure your blood pressure, my observer bias, as the mercury is dropping in the blood pressure measuring device, might influence my interpretation of the results. So subjective measures can be influenced by the expectation of the results. And then we talked about randomization, which is that in any experiment there could be order bias and there could be allocation bias. So order bias is the idea that animals or patients studied at the beginning of your experiment might be fundamentally different to the animals or patients that you study at the end of your experiment. So if you do all of your controls first and then all of your treatments for the second, that order might influence your results massively. And allocation bias is the idea that if a very sick patient comes in, I might put them in the placebo group, and then a mild patient comes in, I might put them in the treatment group because I want to show that the treatment is better than the placebo. Now remember, all of this is subconscious. We don't think that there's massive fraud going on. There is a little bit of fraud going on, but we don't think there's massive systemic fraud going on in science. It's a lot of subjective, subconscious things that are happening. So this is Science 101. This is the basics of science. This is what I would hope every scientist could immediately reel off: the importance of blinding and randomization due to observer bias and the placebo effect, etc. So Science 101 isn't Science 101. So what I'm going to do is I'm going to show some pie charts. Now, instead of using a boring old circle, this is actually a section of a mouse brain stained with maybe crystal violet, maybe toluidine blue, but it's got a beautiful stain here. And this is the mouse brain. This is actually the memory center of the brain. It's called the hippocampus. Isn't it beautiful? But I'm going to use this as my pie chart just for fun. Okay, so in animal studies of neuropathology, so stroke, Alzheimer's disease, etc. In animal studies of neuropathology, what percentage of animal studies is the observer bias blinded? So is the scientist observing the result blinded from which group of animals got the treatment and which group of animals got the placebo, the no treatment group? What percentage are following this fundamental principle that there is such a thing as observer bias, so we need to blind the observer? What percentage? The answer is 20%. So 80% of neuropathology animal studies are not blinded, right? Okay, what about something else? What about randomization? I talked about order bias. It's a huge problem. There's real biological differences between animals at the beginning and animals at the end. There's even been studies that show, say I open up a cage of mice, the first mouse I grab will actually have lower stress hormones than the last mouse I grab. And they largely think it's because the first mouse you grab is out in the open. So it's clearly not stressed, not anxious. So you grab the first mouse out in the open, and the mouse hiding away in the corner, it's probably a stressed mouse, and that's why you picked it up last, because it was hiding and scuttling away. So merely even just picking mice out of the cage, there's differences between the first mice you pick up compared to the last mice that you pick up out of a cage. So we have to randomize. We know that we have to randomize. Study after study has shown that we need to randomize. So what percentage of neuropathology animal studies randomize? 15%. 85% of these studies don't randomize, right? So it's starting to not feel like Science 101, is it, right? Many animal studies, the majority of animal studies are not done in a rigorous way. So what affects this lack of rigor and these potential biases that are seeping into our research? What effects is this happening? Is this having real effects, or is it something that we don't really need to worry about? Another way to ask that question is, do animal models work? And so what we mean by that is, can we use animals to predict whether treatments or techniques or anything that work in the animals, will they go on to work in the humans, right? Once we take animal research to the clinic, will they work? That's the definition of an animal model modeling what we want it to model, which is human disease, human treatment, clinical outcomes. Okay, developing surgical techniques. I think it's quite clear the answer is yes here. Practicing surgical techniques, developing surgical techniques, doing heart transplants in pigs first. A really famous study is there's this deep brain stimulation where you put a thin metal electrical conductor into the brain and you stimulate it, and you can prevent symptoms such as the Parkinsonian tremor. That was developed in a few hundred monkeys, that technique, and has now gone on to be used incredibly effectively in tens of thousands of Parkinson's patients, making real effects. It might even be more, 100,000. I don't know. I haven't been up with the research in a few years. So when developing surgical techniques, a heart is a heart and a liver is a liver. So if you do a liver transplant in a pig and it works, it's probably going to work in a human, those same techniques. Toxicity studies, yes, ish, right? If we do rigorous toxicity studies in multiple animal species, it's very unlikely that when we first inject it into a human, they're going to drop dead, right? On the extreme end of the scale, if we do rigorous animal studies and then we inject into humans, the human isn't going to drop dead. Imagine me saying, I've developed a brand new chemical. I think it's going to cure cancer, but I've never injected it into a living thing ever. And now I want to inject it into you, dear human. You're not going to want that, are you? Right? But I say yes-ish in that it largely seems that rodents are much more robust to high concentrations of drugs. So often we find over 50% of the time when we go into the clinic, we need to drop the relative concentrations, and there's unexpected mild liver toxicity or kidney toxicity. But on the extreme example of it being very poisonous, we don't see that. Developing new drugs to treat disease. Do animal models work for developing new drugs to treat disease? So let's define this. When developing novel treatments, the goal of an animal model of human pathology is to accurately predict whether the treatment will be therapeutic in the clinical setting. So let's look at the data. This is our hypothesis. Let's look at the data and test this hypothesis. So this was a study where they looked at all the chemicals that have ever been tested in animal models for the treatment of cancer. And then they looked at what happened to those drugs following being found therapeutic in animal models. And this is the rate that they found. So this is a rate. For every 1,000 molecules that were found to be therapeutic in animal models of cancer, only a few hundred are tested clinically. Only a few hundred are repeated in further animal models and reach that critical threshold of preclinical evidence to move on to being tested clinically. You can't just do one animal experiment and say, let's go to the clinic. It takes a lot of repeating in order to build up that thesis of information that we're sure that this drug is going to work and then move on to clinically. So only a few hundred out of every 1,000 meet that threshold and get tested clinically. Then only a few go into phase 1 clinical trial, right? And make it through the phase 1 clinical trial. So phase 1 clinical trial, they just inject it into a few adult, typically males, just to check that it's will get into the blood. It won't be toxic. It's well tolerated. It passes those very basic levels of a drug, right? So very few of these drugs get through a phase 1 clinical trial. They go into a phase 2 trial, which looks at a proxy outcome of a clinically relevant outcome. So an example might be, if I give you an anti-inflammatory to treat, say, multiple sclerosis, and then I measure inflammation, I'm measuring a proxy outcome of a clinically relevant outcome. If I measure a loss of motor function, which is one of the symptoms of multiple sclerosis, now I'm measuring a clinically relevant outcome. So very few get the phase 2 clinical trial. And so, and then we get to a phase 3 clinical trial. This is looking a large number of patients at a clinically relevant outcome. So that's movement in multiple sclerosis, survival in cancer, cognitive performance in Alzheimer's disease, these kinds of things. And very few get through the phase 3 clinical trial into FDA approval. It's in fact so few you can't even see it on the graph there. So I'm going to switch it to a log scale. Here's a log scale so we can actually see these numbers. For every 1,000 get through animal models, roughly 300 get tested clinically, roughly 25 get through phase 1, roughly 10 get through phase 2, roughly 5 get through phase 3, and roughly 2 finally get that FDA approval. So 1 in 500 in cancer research of drugs that work in animal models are found and so effective that they end up in the clinic and get FDA approval. Now, this is actually probably a heck ton worse in neuroscience research. So we have essentially not found a therapeutic for Alzheimer's disease, the most common form of dementia, despite finding thousands of therapies that work in animal models. We have maybe one therapeutic for stroke, and we have thousands of therapeutics that have been found to work for stroke in animal models. And so we have one working clinically, but thousands work in animal models. Same with Alzheimer's disease. We have none working clinically, but thousands working in animal models, which is actually why I couldn't draw this graph up for those neurological diseases, because there's so few. We don't know the rates of success. In cancer, there have been quite a few drugs that have now been found to be therapeutic, and so we know a rate of success from animal models. But in neuroscience research, we almost don't know that rate. Now, the one drug that was found to work in stroke probably didn't really come through the animal pipeline anyway. We took it from heart attack therapy. It was a therapy for heart attacks, and we found that it worked in stroke. It's a clot buster, so blood busts a clot in your brain. So even that arguably didn't come through the animal pipeline to stroke. It came through the animal pipeline to heart attacks, and then from heart attacks to stroke. So it's a pretty grim outlook, isn't it? And this is translational failure. So translational failure is the ability for the results in preclinical research to translate to results in the clinical setting, right? Now, what's causing translational failure? Well, there's a little bit of a blame game going on here. Preclinical researchers point to clinical researchers. And why would they do that? Well, let's say I'm a preclinical Alzheimer's researcher, right? Or even a stroke researcher. Let's go stroke. Let's say I'm a preclinical stroke researcher. I say, okay, I've got this result. It's in this very particular kind of stroke, in this very particular location. They are very healthy. It was given at this exact time after stroke, and we found that it worked, right? An example might be youngish, healthy males that we gave the stroke and we gave the treatment 1 hour after the stroke, and we found that it worked. We are happy with our results, and we think this is a great result. They hand it on to the clinicians, and they design the clinical trial. And what often happens, partly because of many of the restrictions of a clinical trial, they give it to any patient that's had a stroke within 24 hours. They give it to women and men. They give it to obese people, people with high blood pressure, high cholesterol. None of this was shown in the animal models. And they give it 24 hours after stroke, 36 hours after stroke, they give this drug. And then they say, look, it didn't work. And the preclinical researcher was saying, yeah, I only told you that it might work in this one setting, and you expanded it to a very large setting, right? The clinical researchers point to the preclinical researchers and say, what is your scientific rigor? It doesn't seem like you have high levels of scientific rigor. Okay, so that's the blame game that's going on. Maybe your models are terrible. Maybe this isn't a model of stroke. You told us it was a good model of stroke. You know, there's something wrong with your field because it's not predicting what's happening in the clinic. Bit of a blame game going on. Let's have an exploration of it. One thing I wanted to say is citizens are really unaware of this. They're really unaware that stuff that works in mice doesn't work in humans. And just an example, I like to read this blog every now and then. It's called Humans of New York. And it's just these really interesting interviews with people on the street. People have interesting lives. And this guy says, I have stage 3 melanoma, which puts me at a 48% chance of survival over the next 5 years. However, I have the ability to speed read very technical material. So I went to the library at Duke and read over 800 papers on melanoma, which doctors just don't have the time to read. And I found a very promising study that suggests chloroquine, there's chloroquine again, combined with the deprivation of certain amino acids, has shrunk tumors in mice to almost nothing. So this guy is now going to go and experiment on himself based off these mouse studies that use chloroquine and reducing certain amino acids in their diet. And he's completely unaware that virtually animal models barely ever predict what's going to happen in a clinical setting, right? And so citizens are really unaware, and that's a really sad thing. And what I think it does is it ultimately undermines science, because every day we read in the paper, we've cured cancer, we've cured Alzheimer's disease, but it's almost always in mice. And I've actually been a part of this. So I published some research about mice using mefenamic acid to treat Alzheimer's disease. And there were terrible headlines all over the world. One said 39-cent pill for menstrual pain cures Alzheimer's disease. That was the headline. Here's another one: menstrual pain reliever drug, mefenamic acid, shows promise as an Alzheimer's disease treatment. And so this went all over the world, and almost inevitably, none of the articles mentioned that the study was in mice, right? They just said, boom, look, we've got this brand new treatment that might work. There were really horrific titles out there. And so one of my favorite Twitter accounts that's addressing this issue is, it's called Just Says in Mice. And all it does is retweets a news article that talks about a discovery, mefenamic acid cures Alzheimer's disease. And all it does is retweet it with the words "in mice," just to raise awareness that a lot of these major breakthroughs that are happening in mice, and mice, this preclinical research is really poor at predicting what's going to happen in clinical outcome. There is massive translational failure in scientific research. So I'm going to explore this in detail to figure out maybe whose fault is it? Is it the clinician's fault, or is it the animal studies fault? I showed you that only 20% of animal studies are blinded, only 15% of animal studies are randomized. So you might guess where this is going. It might be more the preclinical scientist's fault, which I'm one. So I'm trying to improve my fellow colleagues by placing these videos. So in the next video, we're going to do a deep dive on this drug to demonstrate whose fault translational failure is, okay?